stomping on the grapes right here, and then the grape juice comes out here. And there are some yeast that are naturally present on the skins of the grapes, and so they don't have to add the yeast. All they do is take the grape juice and put it inside these clay urns right here, which are anaerobic environments, and then the yeast do their anaerobic respiration, and you get wine out of the grape juice. And that's, this is 5,000 years ago, and really not much has changed. In modern times, they still harvest the grapes, and I think they don't step on them with their feet anymore, at least I hope they don't. But anyway, they put them in airtight casks, and the yeast ferment the naturally occurring sugars, the glucose in the grape juice, and you get wine with alcohol in it after a certain amount of time. Alrighty, any aerobic or anaerobic questions? Alrighty, so this will be the last topic for midterm number 3, and let's jump into signal transduction, which I guess will show up on midterm number 4. Here's a true story related to signal transduction. A couple of years ago, I had, teaching this same class, I had a student named Chris, a really good student, and he eventually transferred to UC Davis. And last semester, he just popped by to say hi, and he said, Mr. Edens, when you gave that lecture on signal transduction, I thought it was kind of dry, but now that I'm taking classes in cellular biology and whatnot at UC Davis, it's the most important thing in the world. Signal transduction is everything. That's actually what he said. So if you think this is slightly dry, bear in mind Chris's experience with it. You may have actually come to appreciate it later on. All right, signal transduction is all about how one cell can communicate with another cell. Every now and then cells need to talk to each other, so to speak, and signal transduction is the process of one cell talking to another. And just to put this in some context inside our own bodies, let me show you what happens when you cut yourself. This is supposed to be a diagram of your skin. Here's the air outside the surface of your body. Here's your skin right here. These are some skin cells there. And this is supposed to be a blood vessel just below the surface of the skin. So everything's fine and hunky-dory. Now you haven't cut yourself, and then somehow you, get in a fight with a beer bottle or something like that, cut your finger, and so now you've got a big gash in your skin right here. Well, the cells in this area want a couple things. They need a couple things. What they need is a little bit of extra blood supply to this region of your body, because they're going to need some nutrients to rebuild themselves, to rebuild the damage, and those are going to come from the blood. So they want a little more blood supply. And they also want some white blood cells, which are the ones that fight infection, to be able to exit the blood vessel, which is where the white blood cells are, to get out of the blood vessel, to go get rid of any bacteria that have gotten inside the cut right there. And so essentially these cells need to communicate with the cells that make up the wall of your blood vessel to say, hey, leak out a little bit of extra blood and send us some white blood cells, or we're going to need that stuff. Well, the way it works is this. The cells that are injured release some molecules called histamines. And histamines have a lot of different functions in the body, but this is one of them. They act as signal molecules to allow cells that are damaged to communicate with the cells on nearby blood vessels. So these histamines just kind of diffuse outward. They're released from the injured cells, and eventually some of those histamines will bind to receptor proteins in the cells that make up the wall of your blood vessel. And you remember, receptor proteins are membrane proteins that allow the cell to detect what molecules are floating around outside of it. And remember that receptors are very specific. They have specific binding sites to bind only the molecule they're supposed to be detecting. So we would say in this case that the histamine is an example of a signal molecule. It's a molecule that moves from one cell to another as a way to communicate some information between the cells. And the receiving cell always has to have a receptor protein for the signal molecule to be able to bind to and detect that particular signal molecule. Now, the cells that detect the signal molecule, that bind the signal molecule, like this one here, they are usually pre-programmed to have some response to the signal molecule. And we call that the cellular output response, the response of the target cell to the signal molecule. And those cells have pre-programmed into their own biochemical pathways the appropriate response to that particular signal molecule. And what is that response? Well, in this case, as we'll see when I click it, is these blood vessel cells right here are going to contract themselves a little bit, make themselves slightly smaller. There we go. And that's good because now this blood vessel gets slightly leaky, so a little more nutrients and things will be able to escape from here and get into the surrounding tissues, and that will give these cells here nutrients to rebuild themselves. And also white blood cells will be able to pass out there and fight off any bacteria that have gotten inside the cup. All right, so the major principle there is that for cells to communicate with each other, they release molecules. One cell will release signal molecules that spread out. The receiving cells need a receptor, and the receiving cells have some sort of cellular output response when they receive the signal molecule. Now, I use the histamine as an example here. Molecules like histamine are called paracrines. Signal molecules that are like histamines are called paracrines, and that means a signal molecule that diffuses between nearby cells. In other words, signal molecules that just react on simple diffusion to come out, to leak out of the signaling cell and just diffuse outward until they get to the receiving cell. Those are called paracrine signal molecules. Here's a different example of signal molecules, but also inside your body. If you are frightened or angry, well, mostly frightened, you've probably heard of, I got an adrenaline rush. And people mean this to see that, when people say that, they mean suddenly they felt sort of almost like an electric surge of energy inside their body, and it feels kind of tingly, and your heart beats and you breathe faster. That adrenaline rush is caused by a signal molecule that used to be called adrenaline, but at some point they switched the name. It's now officially called epinephrine, but it's the exact same thing as adrenaline. And it is a signal molecule. It operates off by the same principles that I just talked about for histamine. Here's a little more details of how it works. Inside your body, actually right on top of each kidney, is a gland called your adrenal glands. And when you perceive something that's very threatening to you, like a monster or something else that's frightening you, your adrenal glands release this epinephrine, this adrenaline, into your bloodstream. And the bloodstream actually carries it throughout the entire body. And so the adrenaline actually goes not just to your liver, as you see here, but it goes to muscles and other tissues and to your heart. Many organs respond to it, but I'm just going to use the liver as one example that's responding to the signal molecule. And same principle we saw before. For the cells, for any cells to respond to a signal molecule, those receiving cells need a hormone that's going to be able to bind to and detect the signal molecule. And just like we saw before, the cells here in the liver have a pre-programmed response to detecting adrenaline and in epinephrine. And in this case, that pre-programmed response is to pump more glucose into the bloodstream. What your body is thinking is if there's some sort of life-threatening crisis going on here, you're going to need some more blood sugar for energy purposes to help you survive the crisis. So the liver releasing glucose is the cellular output response to binding this particular signal molecule, the epinephrine. So it's pretty much the same as what we saw before. The only difference is this, that the epinephrine signal molecule was not carried just by simple diffusion between cells. It was carried in the bloodstream. And so by virtue of that, we call it a hormone. Hormones are signal molecules that travel in the bloodstream, not just by simply diffusing between nearby cells. Yes? Yes, yeah, if somebody is having a severe allergic reaction, like if they eat a peanut, when they have a severe peanut allergy, one of the things the body can do is to cause the trachea, the windpipe, to swell shut, and then the person could asphyxiate because of their allergic reaction. And they have these devices called EpiPens that have epinephrine in them, and if someone's having an attack like this, you can give them a quick injection of the epinephrine, and that epinephrine molecule also binds to some receptors in their windpipe, and it causes the windpipe to relax and they can breathe again. It can save a person's life if they're having a big anaphylactic reaction like that.
The same thing as they stab into people's hearts, right? To get the heart rate, it's the same thing?
It probably is, but the only ones I've ever seen say stab the person in the thigh, but I guess you could put it in the heart also. It might be a little dangerous. Okay, so what I want to do now is zoom in on just one liver cell and look at a little bit more detail of how the liver cell was able to carry out the appropriate cellular output response, which in this case is, as I said, is releasing the glucose into the bloodstream when it binded the signal molecule. All right, so this brown thing is supposed to be one liver cell, and let's zoom in on it so we can see what's going on inside of it. There in the upper left corner is the receptor for the signal molecule. There it is. Here is the chain of stored glucoses inside the liver cell. The liver cells don't store glucose as separate monosaccharides floating around. The liver cells link them all together. Here's a good review question. What do you call that molecule? It's not starch. Glycogen, yeah. It's essentially the same molecule of starch. It's linked together glucoses, but if it's made inside an animal cell, it's called glycogen. If it's made inside a plant cell, it's called starch. Okay, so liver cells are full of glycogen. And to release the individual glucoses into the bloodstream, we need an enzyme that can digest glycogen. So that's what this thing is right here. It's the enzyme that cuts apart glycogen into little glucose monomers so that they can leak out into the bloodstream. And so that is the cellular output response. Remember, in this example is releasing the glucose into the bloodstream. And whatever enzyme, whatever protein carries out the cellular output response, we're going to call the cellular response protein. And it's usually one enzyme or maybe just a couple enzymes that actually do the final response that the cell is supposed to do. All right, so let's watch it in progress here. There's the signal molecule binds to the receptor. The receptor alerts the inside of the cell. Warning, warning, I've detected my molecule. And that leads to the activation of the cellular response protein. And now the cellular response protein is going to carry out the cellular response, in this case, digesting up the glycogen into its individual glucose monomers, which then go out into the bloodstream. Okay, so fine and dandy. Those illustrate two of the three major steps in cell-to-cell communication. There's the reception step, where the receptor protein binds the signal molecule, and there's the cellular output response step, where the cell does the response it's supposed to do, and the cellular output response is carried out by a cellular response protein. What I skipped over was the connection between those two. How did we go from binding the signal molecule here at the receptor to suddenly that cellular response enzyme down here getting activated? So let's go back in time and add in a few more details here. There's the reception part of it. in between the receptor and the cellular response protein are some other proteins that are involved in eventually going from the receptor to activating the cell response protein. And these are called relay proteins. I guess it's almost like a relay game where one person tags the next person, and that person tags the next person, and that person tags the next person. So here's the same pathway again. When you get the reception step right here, the receptor actually physically interacts with the first relay protein and activates that protein. And now the first relay protein then drifts over and activates the second relay protein. And now relay protein number 2 then activates relay protein number 3. And then relay protein number 3 then finally activates the cellular response enzyme, and now you get the cellular output response, the releasing of the glucose. And the whole thing, all the molecules that were involved here, the receptor protein, the first relay protein, the second relay protein, the third relay protein, and the cellular response enzyme, all those proteins together are part of what's called the signal transduction. It's